Good morning. Good morning. Welcome to day 2 of the NBP 18 Symposium. We got off to a fantastic start yesterday looking at the globe, the burden of disease on the planet Earth, and how we can measure it, how we've discovered new ways of measuring it, making those telescopes better, making measurable what is not so. And now we're shifting to another domain, and it's great, and it's a real privilege to have some of the brightest, most innovative people in the room today presenting to you. And it's really gratifying that Denmark is making such a strong contribution to this innovation as well, through iPsych and the other fantastic work. But one of the issues that the field has is how to develop ways to combine information from the genome and information from the environment. And it seems so simple, and many of us live in this 1970s G by E world, which is kind of blazingly simplistic. And now we're trying to find ways to actually get it to be applied in a pragmatic fashion. So what? How do we deal with the fact that how many of our mental disorders are incredibly polygenic? I remember hearing Peter Vischer give a presentation quite a few years ago, where he was talking about height or BMI, and he was saying that it's polygenic, it's very polygenic, it's massively polygenic. Don't get angry with Mother Nature. This is just what she's delivered. And I think that's the attitude. We can't sort of get frustrated or rebel or ignore the evidence. So we live in this omnigenic world, infinitesimal models, Fischerian landscapes. And so what do we do next? And so we need to be very creative in using this information as in our toolkit to invent new ways to leverage discovery. So our first speaker is a good friend of mine, Naomi Wray. She is a named investigator on the Niels Bohr Professorship. Naomi is a professor at the Queensland Brain Institute and a co-director of the program in complex trait genomics at the University of Queensland. This group is large. I think the latest count is 16 postgrads and 20 postdocs. And there's been a fantastic interchange between Denmark and Brisbane. We've had several of Naomi's staff come to Aarhus, and Andrew Schork was from Roskilde, was in Naomi and Peter's lab just a couple of weeks ago as well. And we hope to do more of that in the next 5 years, that creative interchange where we can immerse the Danish staff in the cauldron of discovery that's happening in Peter and Naomi and Jian's group in University of Queensland. Naomi has a particular interest in leveraging the genetic contribution to psychiatric disorders, and she's going to share with us some of her new thoughts and new discoveries. Some of the information's not been published yet, so please don't tweet all of this. But later on, when it's available and in the public domain, we will post her talk and perhaps slightly edited to reflect what is in the public domain. But Naomi, please join us, and we look forward to your presentation.
Well, thanks, John. I'd like to start by saying what a pleasure it is to be here, and how proud we are in Queensland that we have the Niels Bohr Professor, and the way you divide your time between the two groups. And it's a particular pleasure to be here in Aarhus. I have many collaborators over many years, and it's just really nice to be visiting you all, and we're going to be here for a whole week, so I'm looking forward to those discussions. So anybody who looked at the program of the symposium will realise that yesterday was very heavily psychiatric epidemiology, and today is very heavily psychiatric genetics. And I think it's really fantastic that we've got those two groups together, because a lot of what we do in psychiatric genetics is built on the clues from psychiatric epidemiology. But I also realise in psychiatric genetics there's a lot of jargon, and so since I've got the hour slot, I thought that I would spend some of my time kind of trying to make a gentle link between the two. Well, gentle, you tell me if it's gentle. So I think the idea is if there's anything that's not clear, maybe shout out. Please ask questions as I go along, because there's nothing worse than losing the thread early on. So yesterday we heard a lot about comorbidities, which is measuring things in an individual. And I guess those of us who are interested in genetics then try and take that one step further and say, can genetics tell us anything about those comorbidities? And so if there's something genetic, then that means that we're not measuring things in individuals, we're then looking at measuring things in families. And that's the same for if we're interested in environmental risk factors. Yesterday we heard a lot about environmental risk factors of psychiatric disorders. If we want to understand about a genetic relationship, we'd also then have to be measuring families measured for the disorder and the environmental risk factor. So genetic epidemiology has been a very long-standing field, but to be honest, it's difficult to collect the data sets, and so there's been limitations in what we can do. But in the last 10 years, with the advent of the genome-wide association studies, we've actually got a whole new field arising, which is really genomic epidemiology. And so those are the steps that I'm going to go through. So first, in contrasting epidemiology with genetic epidemiology, we heard yesterday about increased risks in disorders. And so from another way to think about that is about a phenotypic correlation. And so to measure that, you're measuring the increased risk of disease 2 in those with disease 1. And when you measure those increased risks, there's many, many causes for comorbidities. I've highlighted a paper there that goes through the many causes. So as I say, one cause might be from a genetic contribution, and so for estimating a genetic correlation, we then have to look at co-segregation in families. And so that means we're wanting to measure the increased risk of disease 2 in the family members of those with disease 1. And in order to benchmark that, that means that we also have to measure the increased risk of disease 1 in the relatives of those with disease 1, and also the increased risk of disease 2 in the relatives of those with disease 2. And so that information is what helps generate the estimates of heritability. So heritability we define as the proportion of the variation in population that is attributable to genetic factors. The trouble with these co-segregation type studies is that it's very difficult to collect large enough cohorts of families. And when you take account of the fact that some diseases have late onset and all sorts of other confounding factors, these samples are difficult to collect. So where we have made progress is in either twin studies or actually with the Scandinavian registries. And so this is a paper, this is using, showing the way in which Danish data have been used to look at the genetic contribution to psychiatric disorders. So this is a paper published in 2010. On the x-axis we've got age of follow-up, and we're looking at children of parents who have or don't have psychiatric disorders. So the bottom line is the cumulative incidence of the psychiatric disorder in those whose parents don't have a psychiatric disorder. This is the cumulative incidence when one parent has a psychiatric disorder. And this is looking at schizophrenia here. And then this is the incidence of schizophrenia when both parents have a psychiatric disorder. So you see, this paper was really about showing how having two affected parents generates this massively increased risk of the disorder. And then the top line was then not just focusing on schizophrenia, but schizophrenia-related disorders. And then here we have the same sort of information, but for bipolar disorder. So the cumulative incidence of bipolar disorder basically in those whose parents aren't affected, in those who have one parent affected, those who have two parents affected. And so I read this paper and I then was thinking, well, is this actually what we expect from our standard genetic model? So under standard genetic model, we say that the genetic factors are additive, and we would, what would we expect under a standard quantitative genetic model where genetic factors are additive? What would we expect if two parents were both affected? So I took these data, just the summary statistics, and from those summary statistics you can actually make an estimate of the heritability. And so this is then, as I say, applying the standard genetic models. Oh, let me go back. Applying the standard genetic models to those increased risks, so massively increased risk of having two parents affected compared to one parent affected, comparing it to not having a parent affected. And the estimate of the heritability was 0.67 from one parent, 0.66 from two parents. I then did the same for bipolar disorder. The estimates of heritability were 0.61 and 0.66. And in the data, they also had information on major depression. And so then the estimates were 0.31 and 0.36. So the take-home message was, well, firstly, it's very hard for us to judge risks to relatives because they're on a scale which is hard to actually gauge. And in advance, before doing the sums, I didn't know what would come out, but the sums showed that actually what we're observing is totally consistent with our models of polygenic disorder. So, and from those data, we can also estimate a genetic correlation. So the genetic correlation between schizophrenia and bipolar disorder from the data they provided was 0.47, and between schizophrenia and major depression was also 0.47. So this was looking at the increase, this is based on data which is the increased risk of bipolar disorder in the children of those who have schizophrenia. But of course, a big caveat there is that I like to use all the time the words consistent, because consistent means that we realise that we're modelling things and we should never lose track that models maybe don't reflect everything about nature. And of course, families share more than just genes. And with these data, it's very hard to separate out non-genetic factors from genetic factors, and maybe things are confounding. Although the fact that we got the consistent estimates would speak against that. The Swedish data, Scandinavian registries, have also been used to estimate heritability for schizophrenia and bipolar disorder. And this was with 9 million national records, 36,000 with schizophrenia, 40,000 bipolar disorder. And in this data set, they had parent-offspring combinations, full SIB combinations, maternal half-SIBs, paternal half-SIBs. So with that design, it's actually it actually provides a way to separate out genetic from non-genetic factors. And so then they made their estimates where the heritability for schizophrenia was 64%, bipolar disorder 59%. But the little dark blue bar there is the contribution which is attributable to shared genetic family factors. And you can see that that actually is quite small. So that's actually our best information across all psychiatric disorders from psychiatric epidemiology, is from these Scandinavian registries. And the genetic correlation here was 0.6. Of course, those of you who, many of us often quote the heritability of schizophrenia, and usually we say the heritability of schizophrenia is 0.8, heritability of bipolar disorder is 0.8, and those are based on estimates from twin studies. And my feeling is that those estimates are actually slightly inflated, and in a way I think that these estimates from the national registries are more robust. But the bottom line is that whatever it is, it's high, and benchmarking, I think, is actually the key thing, not worrying too much about the point estimates. So moving on from epidemiology to genomic epidemiology, the starting point was the genome-wide association study. So this is a very simple design. I'm sure many in the room know about this, but just for the few who feel that they're definitely on the epidemiology only side, I'll just quickly go through it. So we take a case-controlled sample and we genotype them on this chip, which, for intents and purposes, we're saying would measure, say, 1 million places that vary in the genome. And we measure that on all those people, and it's a very simple design. We just look at each of these DNA variants one at a time and just say, is one of the variants, in this case C, more common in the population than T? And we do that for all of those 1 million DNA variants, and we end up with a plot of the tests of association. So here we've got chromosome and position on the chromosome on the x-axis, strength of association on the y-axis. We've got the line where we can declare significance, and that's corrected for this massive amount of multiple testing. And so everything above the line is then significant. And this plot itself was from the Psychiatric Genomics Consortium 108 loci paper. So this is then a timeline of discovery showing back in 2006 we didn't have any replicated genetic variants associated with schizophrenia. In 2014 we had 108 loci, and this number is now over 200 with the data that's in the pipeline. So the key take-home message is that epidemiology modelling I was talking about with the registry data was based on quantitative models where we had an assumption about polygenicity. These genome-wide association studies have provided the empirical data which demonstrates unequivocally that these disorders are polygenic. And at the beginning of the week, many of us here were at a meeting in Copenhagen, which was about how we take forward these results to understand new biology, how to take those results forward to new drugs. And very much a lot of excitement about the new doors that these results are opening in terms of our understanding of disorders. But the key thing is that the disorders are polygenic. And we use the word polygenic, we think about what's happening in a population, but it's also important to visualize what that means for an individual. So I like to show these cartoons which help just visualize polygenicity for a person. And so each of the big squares I'm calling a genomic risk profile. So it's someone's risk profile from their personal genome. And I'm saying, let's imagine disease is underpinned by 100 risk loci. And so these big squares have got 100 little squares in them, and I've coloured those squares white if the person has no risk alleles at the locus, blue if they have 1 risk allele, and red if they have 2 risk alleles. And in this example, I've said that risk alleles have a frequency of 10% in the population, and the disease that underpins this has a heritability of 50% and has a frequency in the population of 1%. And so then I'm saying these people are the people that are cases, and these are the people that are controls. And the count of the number of risk alleles is above each square. And so the key take-home message is this visualization of, firstly, cases have, on average, more risk loci than controls, but that every single one of us is carrying risk loci. It's just that we don't have a high burden of them. And the other key message is just to get a visualization of the actual heterogeneity between people. That we talk about polygenicity. It sounds very easy, it's very easy to model, but in terms of each individual, it actually is reflecting very different combinations of variants between people. And that may be consistent with what we see in the clinic, with the kind of heterogeneity at clinical presentation. So one of the things that people who are not in the field often find hard to understand is why we need such massive data sets. And to explain that, I use this toy cartoon. So now I have the same thing. I still have my 100 risk loci which are associated with disease, but I'm saying, well, not all risk loci in the genome are associated. So I'm now embedding them in a total of 400 loci, so 3 of them are not associated. And I've also coloured these homozygote, heterozygote, homozygote, but they're not associated with disease. And you can see from these visualizations just how difficult it is to pick out the loci that might be to do with disease. And that's why we need large samples, because we have to separate out the wood from the trees. So polygenicity for me is very, very consistent with the field of network biology. This is one of my favourite papers. It talks about robustness. It doesn't use the word polygenic at all, but all the concepts in there are totally consistent with the way I think about polygenicity. It's saying that we've got biological networks, which when we alter a particular point from a DNA contribution, that system is very robust. There's many different routes around having a perturbation. But it's only when there's a high load of perturbations that you might get with someone who's vulnerable to disease, that the network becomes more vulnerable and more open to, more unable to compensate. Okay, so as I say, there's now a whole field in taking forward the results from genome-wide association studies into what I'm calling genomic epidemiology. And in genomic epidemiology, there's a couple of concepts that we use, and you're going to hear in the other talks. And one is the concept of SNP heritability. So SNP heritability is when we take our data from our genome-wide association study and say, how can we summarize this information? How much more similar genomically are cases compared to controls? And we come up with a statistic which explains how much variation in the population is attributable to those genetic factors in total. And so we call that estimate a SNP heritability. So just like a normal heritability is the proportion of variation in the population attributable to genetic factors, a SNP heritability is the proportion of variation in the population attributable to the genetic factors that we've measured in our study, and those are genome-wide. We know that our studies don't capture everything. And because we're using an awful lot of data to measure one statistic, we can measure that pretty accurately from our data sets. And the estimates that we get aren't really going to change as sample size, aren't expected to change as sample size increases, other than with respect to the standard errors. But the other thing that you're going to hear people talking about is polygenic risk score prediction. And there's many different ways in which that's called. It's called polygenic risk score prediction, it's called genomic profile prediction, it's called genetic risk scores. They're all the same thing. And for that, we take a genome-wide association study, which we call our discovery sample, and we find the estimates of all the effect sizes of all the DNA variants that we've tested. So remember, we don't know the true ones, we're testing everything. And then we take those estimates of those DNA variant effect sizes into a new data set. And we use those effect sizes, and we simply really count up how many risk alleles the people have in this new data set. In this new data set, we also know whether people are cases and controls, and we evaluate those profiles which are made on those people and say, how well do we separate case controls based on these risk profile scores? And so, for example, for schizophrenia, based on the 2014 data, the estimate of SNP heritability for schizophrenia is about 25%, and the outer score prediction from this genomic risk profile scoring is 7%. And one thing I find is that people get confused about why these estimates are different. And the reason they're different is because we've got error in the estimation of our SNP effect sizes. As sample sizes increase, we are going to get better and better at estimating those effect sizes, and this number is going to go up, and we would expect it to approach this number. I estimate that we'd need about 250,000 cases to get our predictions explaining this much variance. But already in height, so often in complex trait genomics, we use height as a model trait because for many reasons, the data sets are larger, we get it's a simpler trait to understand. And with height, we are getting already much closer to the outer sample prediction compared to the SNP heritability. So we can also take, and this is the exciting bit for genomic epidemiology, is that we can take the results from more than one genome-wide association study. So we can take the results from 2 genome-wide association studies. So we have what we call the summary statistics, which are just those effect sizes estimated for each DNA variant. And then we can say, are those effect sizes similar between the 2 disorders? And if they're similar, that's telling us something about the shared genetic information between the disorders. And this is a really exciting paradigm because you then get these data sets, for example, schizophrenia cases and controls, bipolar cases and controls, totally independently collected. We can make sure that there are no relatives, no close relatives between these data sets. And so we get around this potential confounding with environment. And so from these data, we can estimate genetic correlation. And I was leading the study which first applied this to, and it was first applied in psychiatric disorders. And for that, we used the full available data, that is, the individual level data. But these days, we can just take the summary statistics to get good estimates of correlations between data sets. So this is a bit of an old slide from a review in 2014, but it just gives an overview. So this is looking at different psychiatric disorders, and the red bars are the estimates of heritabilities from family studies, and the blue bars are the estimates of SNP heritability. We can talk later about why the differences exist, But the key take-home message is for basically every disorder that we've looked at, we can demonstrate that there's variation explained by common SNPs. All the psychiatric disorders are looking like they're polygenic. Autism may be an except. Well, that's a whole talk in itself, but we'll talk about that later. This is also an old slide, and it's showing estimates of genetic correlations between psychiatric disorders, so different pairs of psychiatric disorders. But the reason I chose this slide to show is because I've got two different colours here. So the light blue are the estimates of genetic correlations between pairs of psychiatric disorders, and the dark blue ones are estimates of genetic correlations between pairs of data sets which are of the same disorder. And you don't see these estimates put out there very often, but I think that they're really important because they're like a benchmark of knowledge about our data. That obviously our expectation is if we take one schizophrenia dataset and another schizophrenia dataset and we estimate the genetic correlation, we expect it to be 1. If it's not 1, then that maybe is telling us something. And I've worked a lot with the psychiatric datasets. My feeling is that the estimates between the schizophrenia datasets tend to be close to 1. The bipolar datasets tend to be a bit lower than that, and the major depression lower still. And I think that's telling us something about heterogeneity. So these days we can go to a website called LD Hub. On an LD Hub, they provide estimates of SNP heritabilities from every genome-wide association study that they can get their hands on. And they've also used the UK Biobank. So the UK Biobank is this community sample of 500,000 healthy volunteers aged between 40 and 60, measured for a whole bunch of phenotypes. And so I just went on there yesterday and I just picked out a few of these just for example's sake. So qualifications, college or university degree, SNP heritability of 0.28. Now Peter's going to be talking a lot about educational attainment later on. Body mass index, 0.23. Age at first live birth. So this is the age you were when you had your first child, and that's showing a high genetic contribution. Of course, you then start entering into thoughts about, well, what is a trait? What are all the factors that contribute to a trait? That's not just about reproduction, it's about personality, lifestyle. Smoking status, 0.15. Unable to work because of sickness or disability. Cycling to work. Financial difficulties. Meat eating. And so you suddenly realise that things that maybe we measure as environmental risk factors can are complex in their own way. You can also go to LD Hub to look at estimates of genetic correlation. So again, they've loaded up 196 traits. And so I just pulled out all the correlations with schizophrenia, which were greater than 0.1. There were about 20 that had correlations greater than 0.1. And the first takeaway thing is to realise, well, out of that 20, there's a whole bunch which make sense, that they kind of fit with our expectations, that the highest correlation is with bipolar disorder, other high ones with depression, sleep duration. So schizophrenia, the genetic profiles, risk profiles for schizophrenia are genetically associated with needing to sleep longer hours. So just to re-emphasise, for those who aren't familiar with these sorts of data, is that these data sets are totally independently collected. So data sets on sleep duration will be from community samples, which people with schizophrenia will be maximum of 1%, most likely less. This is just using natural variation in the population and saying how the genetic factors for those traits how similar they are to the genetic factors for schizophrenia. We've also got neuroticism, openness, anorexia, ADHD, autism. Again, recapitulating a lot of our expectations. Chronotype. So that's saying that people who are owls, who like to stay up more late at night, their genetic factors that underpin that are associated with those for schizophrenia risk. So once you see that there's things in there which make sense, you then start taking notice of the things that are maybe more surprising. So here was one, chronic kidney disease, genetic correlation minus 0.2. But then you have to have a little word of warning. So I then went to see, well, what actually is the SNP heritability for chronic kidney disease? And it was actually very, very low. And so I think you have to take that into account a little bit when you're considering this. I think it's still worth thinking about because it's still an estimate, it's still significant. But when the SNP heritability is very low, for me it's less interesting than when there's a big genetic contribution in its own right. But then you look at the other things which are maybe more surprising. So there's a correlation between ALS, motor neuron disease, and schizophrenia. And I also work on motor neuron disease. The community are very interested in this result. And we've actually replicated it in Chinese samples, so independent samples. Citrate, well, what does that mean? Biologically, maybe that's giving us a clue. Lupus, I can't say that. Inflammatory bowel disease. Yeah, that's maybe then pointing to some of the inflammation hypotheses for schizophrenia. Okay, but then you start thinking to yourself, well, what actually is a genetic correlation? And I suppose when we think about a genetic correlation, what we're thinking is that we've got a causal DNA variant which is impacting two phenotypes, and so that's what we call pleiotropy. But maybe there's other things going on. So one of the things, and this was something I was very concerned about when we did our first analyses, was is it possible that the reason we're picking up a correlation is actually because of some sort of misclassification? And that's something to think about. Maybe it's actually, we're picking up a correlation, genetic correlation, because the genetic risk factor is actually causal for one, and then there's this phenotype is causal for the other phenotype. And then maybe there's some biological pleiotropy going on. Maybe there's two different causal risk factors which are both detected by the same genetic variant that we measure. And this, of course, may also be interesting in its own right, because although it's saying different causal variants, it may actually also be pointing to the same genes. And so that's still interesting. And then you start asking your question, well, a genetic correlation, would we expect to have the same correlation across the genome? And so if you estimate a genetic correlation of 0, does that actually mean 0 everywhere, or does it mean a positive genetic correlation in some parts of the genome and a negative other parts? So I'm first going to just have a look at this misclassification issue. So as I say, this was something I was worried about in our first analyses, and so actually did an analysis of this prior to the publication of the first paper so that I could feel confident that the estimates that we gave made sense. And so schematically what I'm saying is we've got a genome-wide association study of cases and controls. And that's what we think we have. But what happens if, in fact, this is what we've got, that a proportion of our cases have actually been misdiagnosed as cases from here, and perhaps also that we've got misdiagnosis in this direction. We can also think about misdiagnosis in one direction only. And so I didn't really have a feel of those results, and that's why modelling helps look at that. And so this was the results of the modelling. So this is now looking at the proportion of misclassified cases. In this example, I'm saying that the misclassification was going in both directions. So I think that's maybe true in schizophrenia and bipolar disorder. People maybe have a first diagnosis of one and swap to the other, vice versa, what is actually their long-term diagnosis. And then the lines are the true genetic correlations, and then the different colour lines reflect different true genetic correlations, and here on the y-axis is the genetic correlation that we would estimate. So the red line, in a way, is the most interesting one. We're saying the true genetic correlation is 0. What genetic correlation would we estimate based on different proportions of misclassification? So obviously, with no misclassification, we estimate it as 0, but as that proportion goes up, we estimate it to be non-zero. But interestingly, if the true genetic correlation is actually very high, then misclassification between the two disorders actually has less impact, because they are already genetically related to each other. So what's the likely outcome for the diseases that we're interested in? So, schizophrenia, bipolar disorder, I think misclassification estimates have been up to 10%. I think in our data sets that were in the genome-wide association studies, that might be on the high side. But even if that were true, even if the true genetic correlation was 0, the estimate we would expect to get would be about 0.2. The fact that we estimate something that's about 0.6 means, I think, that there is definitely true genetic correlation. I've brought this in as, again, with all these estimates, you always have to try and think outside the box, think of things that could go wrong. This is an example which came about of a conversation when Andrew was visiting us a couple of weeks ago. And it's a conversation with Wouter van Rienen, who's a postdoc from Utrecht, who was also visiting at the time. And we were talking about the impact of having super clean controls. And in psychiatry, we do that quite often. If we're studying schizophrenia, we might take out of the control set everybody who's got any psychiatric disorder. And Wouter was telling us how, in his experience with working on motor neuron disease, in a genome-wide association study of motor neuron disease, which is a late onset disorder, so they had older, healthy controls. And because they were older, healthy controls, they were actually depleted of ApoE4, which is associated with Alzheimer's disease, and so they ended up with an association of ApoE4 with ALS. And so you always have to think about these things. What actually are our data sets made up of? So then that got us talking and say, well, what is the impact in our genome-wide association studies of making super clean controls? Again, we didn't really have a feel, but again, you can do the sums to have a look at that. So this was the example of schizophrenia and depression. And we're looking at the impact of super clean controls on heritabilities and on, which is the green line and the red line. And then the impact on the genetic correlation. And the outcome of that is that actually we felt like it actually doesn't actually have that much impact, nothing to worry about given everything else that goes on in our assumptions and modelling. But we did come up with an example where we thought it did have an impact, just to show that it could have an impact. This is an example of hay fever and asthma. And if you were to clean out all the hay fever cases from asthma and vice versa, you would could massively overestimate genetic correlation. So I've talked about this, so I'm now going to talk about this. How do we separate out this pleiotropy from potential causality? And this is, we can do that now with this method of Mendelian randomization. So I'm not really an expert on Mendelian randomization, but I do think it's a useful tool. So the basic concept is that we can compare a randomized controlled trial with Mendelian randomization. In a randomized controlled trial, we randomly allocate people to have a clinical intervention or no intervention, and then you measure the outcome of interest. And we try and set things up so that the confounders are equal amongst the groups. So Mendelian randomization then says, well, people's genomes are randomly allocated to them with respect to many things that they have in life. Can we use a genetic variant as the equivalent of an intervention compared to the other allele? And then measure outcomes of interest, and that expecting that environments aren't predestined by the alleles people are carrying. So Mendelian randomization is borrowed from economics, from instrument variable analysis, and I think there's many people here who would understand that better than I do, but my simple way of understanding it is to say that in economics, the sorts of things they might study is what happens to if you increase taxes on, say, smoking or alcohol, a big event which you can measure and know about, you can then look at the impact of things downstream given that intervention, because you know that increasing tax may decrease usage for a while. Or if you add an extra year of schooling in a schooling system, what impact does that have? And so this then borrows this methodology to say, if we've got something that we know is associated if we use the genetic variant as this instrument. And so the thing that we're actually interested in is we're wanting to measure the relationship between an environmental exposure and an outcome disease. This is what we want to know, but we haven't been able to measure that. And even in a randomized trial, it might not be ethical to measure it. But what we have is the relationship between a DNA variant and that exposure trait, and the relationship between that DNA variant and the disease trait. And the maths to estimate the thing that we're interested in is complicated, but the answer is very simple. And it turns out you can estimate this, the relationship between the exposure and the disease, just as the ratio of the two SNP effects from the two traits. So this might be the SNP associated with major depression and a SNP associated with, say, body mass index. And from those two genome-wide association results, we can get an estimate of the relationship between the body mass index and major depression. So I was never a very big fan of Mendelian randomization because when people first talked about it, we didn't have many SNP variants associated, and the SNP variant effect sizes weren't very big. But now things have moved on. We have many SNP variants, and in fact, I think it does become a very powerful tool because you can then say, we can measure this for one associated SNP, and then we can measure it for all other associated SNPs, and then we can see, when we measure it multiple times, do we get a consistent result? So this is a sort of plot you might see. It's saying, here's the effect size of the SNP for the exposure trait. And we take that to be genome-wide significant, so there's a big gap in the middle. And then we estimate the effect size for those DNA variants for the disease trait. And we're looking to see if this ratio of the estimates is the same, which is equivalent to having a look at the slope of the graph. And you can see in this toy example, pretending I've got quantitative traits which are standardized, and then you would realize that the effect sizes would be much bigger for the exposure trait, but and smaller for the disease trait, but still it's the ratio that could be interesting, because obviously the exposure, the effect may be diluted down, but it's really that we're seeing a consistent effect. So basically, we would do this in traits which we see there's a genetic correlation, and if that genetic correlation was driven by pleiotropy, we wouldn't really have any expectation that the effect sizes would be correlated between the two traits. It could be that both variants are associated with the two traits, but there would be no expectation that one would have a higher effect, that there'd always be a relationship. But under causality, that's what you would expect is a relationship. So there's many variants of Mendelian randomization. They all essentially do the same thing. It's all about, the different versions are really about trying to maximize power. We like our method, which was published this year in Nature Communications, and this is work led by Zhan Yang and Zai-Hong Zhu. And what's nice about the method that they have put forward is that it takes account of the errors in the estimates of the SNP effects, and so that gives you a little bit extra power. And the other thing that it does is it checks that the results that you get aren't driven by exceptional pleiotropy. By looking for outliers. So what do I mean by that? This is an example in the paper where we look at the relationship between LDL cholesterol and Alzheimer's disease. So we're taking the SNPs that are genome-wide significant for cholesterol, and then which is the effect size on the x-axis, and then we're looking at the effect sizes for Alzheimer's disease. And then we're calculating our BXY coefficient, and you can see if we apply it blindly, we get this estimate of 0.3, very, very significant result. But you can see we've got this massive outlier. Anyone know what that massive outlier is? ApoE, yeah. So ApoE4 is very highly associated with Alzheimer's disease. It's to do with lipid metabolism, and so it's associated with LDL cholesterol. So this is a true example of pleiotropy. You've got the variant which is having effects on the two different disorders. If you get rid of that, because it's such an outlier, then we get no association. This is an example just to show that it doesn't always make things less associated. This is an example of BMI and type 2 diabetes, and the effect size actually became more significant by getting rid of the outliers. So it's not a one-directional thing. So I'm now going on to have a look at how we've applied Mendelian randomization in our latest PGC paper from the major depression group, where we identified 44 loci associated with major depression. So these are estimates of the genetic correlations with major depression. Again, many of the things which make sense based on the talks from yesterday: body fat, body mass index, coronary artery disease, triglycerides, and then psychiatric disorders down here. So in our Mendelian randomization, a lot of people would take forward all of these two Mendelian randomization analyses, but we just took forward a few based on epidemiology. So we looked at body mass index in coronary artery disease. That fits very much with Moretta's talk yesterday. Years of schooling, there's been a lot of debate about the direction of effect between years of schooling and depression. And we kind of took schizophrenia as a kind of benchmark control. Not that we're expecting causality, but just to understand what the analysis would do in a situation with a correlated disorder. So these were our results. So we took body mass index as being causal for major depression, and there were 85 genetic variants. I'm using the word causal like this, okay? Taking 85 SNPs, which are genome-wide significant for BMI, and we get a very significant result. But in contrast, if we take the SNPs which are associated with major depression into the BMI data set, we get a result which isn't significant. And similarly, education is as causal for major depression, very significant in one direction and not the other direction. Coronary artery disease, with these analyses, were underpowered. And so that needs more data to understand that. So do I think these analyses are useful? Well, I think they're useful because they're so easy to do. And they add a little bit of information. And I almost think that the non-significant results are more interesting than the significant results, because the non-significant results, I think, do give us a reason to say that there isn't a direction of causality in that direction. The significant results give us something to think about and not use the word causality, but just think about all the things that could be part of a relationship dynamic. Because there are many assumptions under Mendelian randomization. I'm not going to go through them here, but the assumptions are all really about possible confounding factors. When we applied the methodology to schizophrenia, so using the 105 SNPs significant for schizophrenia, very significant result. The 36 SNPs significant for major depression, very significant result. So this is really showing pleiotropy, really true biological pleiotropy, true shared pathways, very much consistent with shared biological pathways between disorders of the brain. I'm an associate editor for JAMA Psychiatry, and to be honest, we're getting overwhelmed with Mendelian randomization analyses, and they're all, getting to be a little bit dull. And they're the sort of thing, I feel really guilty hearing all the talks of yesterday, the years of work go into those studies, and then people will run this analysis in an afternoon, write it up. So we then wrote this kind of editorial, which was really about some guidelines. And basically, the guidelines say, don't bother to send us a Mendelian randomization study unless you've got 10 genome-wide significant SNPs in both directions, because I think it's important to be testing in both directions. And even then, to be honest, I don't really want to see one subject at a time, bundle it up. That said, we have done a Mendelian randomization between schizophrenia and breast cancer. So this is the one that we were talking about yesterday. And you can see 150 variants going from schizophrenia to breast cancer and 204 genome-wide significant from breast cancer. And we're getting a significant result for the Mendelian randomization in this direction and not in this direction. So I do think that's a useful result. This came up yesterday, and I think from this, it's a take-home message is that women with schizophrenia should be encouraged to take part in breast screening. Now, John wanted me to talk about environmental risk factors, and so I don't feel like an expert on this at all. And my only foray into it has been with childhood trauma and depression in collaboration with Wouter Peiro, who's a very talented, he was a PhD student when he worked with me, but is now a postdoc. He's a psychiatrist as well. And so before he came to work with me, he'd already published a paper about the effects, the relationship between childhood trauma and depression using polygenic risk scores. Yeah, childhood trauma, these are just some of the estimates from the literature that childhood trauma, so this could be many, many things, parental divorce and all other sorts of, and then we see these are the sorts of odds ratios, increase odds of depression for those who have trauma compared to not trauma. So, really kind of massive effects. And, many of our studies ignore this altogether. And so it is, I think, important to be able to think about how to analyse data if we have data available, to look at the relationship between a genetic and environmental risk factor. So in this first paper, he showed a genetic environment interaction. He then went on and did a bigger study which showed no genotype by environment interaction. Jerome has been working on a paper using the UK Biobank, and Wouter and I were in big discussions with him about that. And now Wouter is writing a paper which is saying, we've thought an awful lot about G by E analyses. The more we think about them, the more complicated they are. These are the lessons we've learned, so hopefully other people can build on the lessons, but it's a field that the more I get into it, the more I feel I want to get out of it. So just one other thought. Again, as I say, when we get results, we always have to think out of the box. What could be driving them? What artifacts could be driving them? There's this paper by George Davies-Smith and Marcus Manafo talking about collider bias. Again, that's something that in epidemiology you'll be very familiar with. In genetics, we tend not to have thought about it so much. But this paper was saying in the UK Biobank, could there be any, we have to recognise this isn't a population sample, which is one of the advantages of the studies you have in Denmark. This is a community sample of volunteers. And so, for example, I was thinking, what happens if people with depression and obesity are less likely to join the study? How could that generate biased estimates of correlations? In summary, I think that genomics data has really opened up our ability to study relationships with environmental and other risk factors in a way that really wasn't possible before. And it really is a byproduct of having the available data. It's not that we're setting out to raise money to do these studies. The data are there and we are able to do them. It's very quick often to do these sorts of analyses, but I do think the interpretation can be tricky and we have to be a little bit cautious and think out of the box. I think null results can be valuable. Oh, and I put that in there because I get fed up of seeing papers which put things with more than 2 decimal places, because that implies that people think that there's a greater confidence in our estimates. There's so many assumptions going in, no more than 2 decimal places. So in common conclusion, thank you to my funders, thank you to our group, all in our height distribution, wearing our T-shirts. Particular thanks to Peter and Jeanne, who lead the group with, and all the discussions with them, which really helped shape my thinking. And finally, plan ahead for those who think about quantitative genetics. The International Congress of Quantitative Genetics is coming in 2020 to Brisbane, and I know it's a long way, so plan ahead. Thank you.
We've got time for questions.
Conveniently, no time for questions.
Do other people find that photograph a bit creepy? It looked like a military parade. I also want to quickly jump in and say, for the students who heard Naomi say that she thinks that the more she thinks about gene by environment interaction, she wants to get out of the field, she was having a joke. So please don't leave the field. She's feeling exhausted, but you are young and energetic.
I think they're very interesting questions. But I think we shouldn't underestimate the complexity, and that's the purpose of a Niels Bohr Fellowship,
exactly.
to tackle these hard questions.
That's good. We have some time for questions from the audience. Anna, have you got your hand up there, or you're just pointing? Questions from you?
Good.
Anna.
Thanks, Naomi. That was a beautiful talk. So my question is, how are you guys thinking about using instruments so SNPs that have high population differentiation and as instrument variables? And are you concerned at all about those SNPs also having other sort of environmental factors that are correlated and could predispose to health issues?
Yeah, I didn't expect an easy question from you. I don't know the answers to those questions. I think they're interesting. Yeah. Well, I think that, admixture is one of the next big challenges. At the moment, we've not really worked with the admixed populations. They've been kind of chucked away, but now we're getting large data sets. Those are going to be more and more interesting. Maybe those are the data sets to focus on. We all worry about population stratification, but of course, sometimes there are differences between populations as well. So, height, for example, there are real differences.
Did there?
Yes, there's definitely population differences there.
A tall Dutchman over on the
yes,
far
it is.
side.
But I'm not going to answer that anymore. We can discuss that at coffee.
Any other questions? Annis?
Thank you very much, Naomi. Brilliant talk, as always. I wanted to ask about the Mendelian randomization, where you see no evidence of directional effects in these, genetically correlated traits, and you kind of use it. I think you used it as a, some kind of warning or some kind of caveat that these might not represent true pleiotropy. Is that correctly understood?
No, I would say, so you can have a genetic correlation and, I'm not really an expert, but you have a genetic correlation and then the Mendelian randomization could be non-significant in both directions. And I think that then that is then saying it still could be that the ones that are positive are also have a positive effect, but there's just not a relationship between the biggest one and the smallest one of those instruments. So that then, to me, is really true pleiotropy, because they're variants which are affecting both disorders, hence you're getting the correlation, but it's showing not causality.
Right. And how does that, just one small addition to that, do you think that the fact that you're actually only measuring the genome-wide significant hits in the Mendelian randomization analysis, while genetic correlation, you measure the entire, all the variants. Does that would that influence the way you interpret these results as well?
Well, I think that's the point of Mendelian randomization, is to take instruments that are truly associated, and so I don't think that's an issue. I don't know the same answer.
Carsten and then Peter.
Oh.
Thanks, Naomi, for a brilliant talk. I also like your paper on this Mendelianization on best practices. Could you also write a best practice on how we do gene-environment interactions? I know it's a tricky question, but there's so many things going on. What timing and all these genome-wide association studies, they assume there's no effect of... there's no gene-environment interaction, so I think it's difficult to go ahead. So it would be good if somebody like you could give some...
So that's actually what Wouter Perot is doing. He's taking the lead on writing, it will be a review article, and Matt Keller is involved as well, because Matt Keller is someone who also has thought a lot about genome-environment interaction. Okay. And I think the way it's going to end up is not, I think, a best practice, because it's hard to know. I think we've come away all saying we're a bit confused, but to actually write down the things that we've learned, so that then maybe we can open up the discussion and other people can build on what we're doing. Because I think otherwise there is a lot of naive application of G by E analyses, and we want to help the field not go forward with that.
Peter.
Yeah, thanks very much for your very clear presentation. I was thinking, you presented something about the Mendelian randomization concerning the association between depression and cardiovascular disease. And to me that's very fascinating. And basically what you pretty quickly said, well, there was no effect either side, so maybe that could be preoperative.
I didn't say that. I said I thought it was underpowered. It did look like there was an effect in one direction. It was underpowered, and this is a very good, very good segue into Jerome's presentation. So maybe with that, because we're eating into his time, we should.
But I didn't see that underpoweredness in the standard errors, because they were as big as the other.
But it was the standard errors relative to the effect size, and so in one direction the estimate was actually close to 0, but the other direction the estimate was actually above 0, and so if the effect size isn't very big, then you need to get the standard error down to work out whether it's real or not. And I think that's where we are, and that's what Jerome is going to be following on with.
Okay, you join with me and thank Naomi for a fantastic presentation.